Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine you're walking this kind of medical tightrope.
Okay.
On one side, you've got dangerous bleeding, needs stopping.
Right. Urgent.
But on the other side, there's this fear. If you try to control that bleeding by, stopping blood clots from breaking down naturally.
Which is one way the body heals, yeah.
Exactly. The fear is you might accidentally cause a different kind of clot, a dangerous one forming where it shouldn't, like in your veins.
Venous thromboembolism.
That's the one. And it's been a real dilemma, for doctors, for patients. Sometimes it limits treatment options for people who really need help with bleeding.
It really is a classic medical balancing act. The body's clotting system, it's just incredibly complex. It's designed to, plug leaks fast, but then also carefully take that plug apart when the job's done.
Right, the cleanup crew.
Yeah, and interfering with that cleanup, that's always had this big question mark hanging over it. Does stopping the breakdown somehow, encourage new unwanted clots?
And that's exactly the big question we're diving into today. We've got a stack of sources here, mainly a new health news piece and the scientific paper it's based on, both just out, actually, May 26th, 2025.
So our mission, really, is to unpack what this new research tells us about the body's system for breaking down clots. Specifically, does messing with it, does it truly raise the risk of those dangerous vein clots, venous thromboembolism?
And this is some serious research we're talking about. It's from a team at UNC School of Medicine and international collaborators, too. They used a whole bunch of different methods.
Which is great. Makes the findings stronger.
So let's get into it. How did they tackle this? What was the core idea they were testing?
Well, at its heart, it was built around that old concern. If you lose or suppress the activity of this clot breakdown system, does that actually make people, more likely to get venous thromboembolism?
And the breakdown system involving that key protein, right? Plasminogen?
Plasminogen, exactly. And its activation.
Think of plasminogen activation, like the signal saying, okay, time to dissolve the scaffolding, the repair is done.
Good analogy.
So the question was, if you turn down that signal, do you accidentally make it easier for, new scaffolding to pop up where you don't want it?
That's it, precisely. And to test that directly, they didn't just rely on one kind of study. They used a multi-method approach.
Which, as you said, is powerful if different methods point the same way.
It tends to mean the finding is pretty robust, yeah.
Okay, so walk us through these different approaches. What were the parts of their investigation?
Okay, first they looked at real-world human data. People who naturally have a deficiency in this plasminogen system.
How? How did they find these people?
They use data from an international registry. It focuses on individuals born with this rare genetic condition where they just naturally have much lower levels and lower activity of that plasminogen protein. Congenital type 1 plasminogen deficiency, it's called.
Ah, okay. So it's sort of natural experiment, people whose system is already turned down.
Exactly. Then next up, mouse models.
Right, the animal study.
They used mice that were genetically modified, so they had either reduced or completely absent plasminogen.
Okay.
And they also took just normal, healthy mice and treated them with a drug, tranexamic acid.
Oh, I've heard of that one. It helps stop bleeding, right?
It does. It works by suppressing plasminogen activation in humans, helps prevent clots breaking down too quickly. So they used it in the mice, too.
And what did they do with these mice, the modified ones and the treated ones?
They used a specific lab model designed to basically trigger venous clotting in these mice to see if suppressing the breakdown system made those induced clots worse.
Got it. Give the mice a situation where clots are likely, then see if messing with breakdown makes it worse.
Precisely. Then moving beyond the animal models, they did large-scale human genetic analysis.
Wow. Okay. How large are we talking?
Massive databases. Genetic info from hundreds of thousands of people. UK Biobank. Data from the INVENT Consortium.
And they were looking for?
They looked to see if variations in the genes, the ones that code for the proteins in this plasminogen activation pathway, if those genetic variations were linked to the risk of getting venous thromboembolism in the general population.
So, checking if subtle, common differences in these specific genes show up as a risk factor for clots across lots of people.
Yeah, and they compared it to genes we already know influence clotting risk, like factor V or prothrombin, to see if the patterns were similar.
Makes sense. A sort of positive control.
Right. And then finally, using data from one of those big human databases again, they looked at actual protein levels.
Not the genes, but the proteins themselves circulating in the blood.
Exactly. They measured the amounts of different proteins from the plasminogen activation system in people's blood and checked if having higher or lower levels was associated with developing venous thromboembolism down the line.
Okay, so let me recap. They looked at humans with a rare natural deficiency, mice with genetic changes, mice treated with a drug, huge human genetic databases, and protein levels in humans.
That's the lineup. It's really comprehensive, covers a lot of ground.
It certainly sounds like it. And you said the findings were remarkably consistent.
They really were, which is what makes the conclusions so interesting.
All right, let's get to it then. The results. What did the science actually reveal? Let's start with those humans, the ones with the natural deficiency.
Okay. So in that international registry of people with naturally low plasminogen activity, here's the kicker.
Yeah.
None of the individuals reported having a history of spontaneous venous thrombosis or pulmonary embolism.
None. Spontaneous meaning out of the blue, not after surgery or something.
Right. And pulmonary embolism, that's a really serious type where the clot travels to the lungs. They just didn't report those.
Wow. That's quite something. People naturally missing a key part of clot breakdown didn't seem to get these dangerous vein clots.
It's a very striking finding. Now, the sources do mention they reported other issues, things like painful growths on mucous membranes, which are linked to this deficiency, but specifically not the spontaneous dangerous vein clots.
Okay, very clear result there. What about the mouse models? The ones where they genetically altered the plasminogen levels?
So in that mouse model where they deliberately induced venous clotting, the mice that had partially or completely lost their plasminogen gene genetically, they did not form larger venous clots compared to the normal mice.
Really? No difference in clot size?
No difference, not at 6 hours after inducing the clot and not at 24 hours either. And the clots themselves, they looked similar amounts of blood cells, similar amounts of fibrin, which is like the main structural protein of a clot.
So even without the normal ability to break down clots, the initial formation, the size, the basic structure wasn't worse.
That's what they found. And then they looked at the mice treated with that drug, tranexamic acid.
Right, the one that suppresses plasminogen activation.
First, they confirmed the drug was suppressing the system in the mice, much like it does in humans.
Okay, so the drug was working as expected.
Yes. And crucially, treating the mice with tranexamic acid also did not lead to larger venous clots. And no changes in the clot composition either compared to the control mice who just got saline.
So the drug that stops breakdown didn't make the clots bigger in that model. Yeah. Consistent with the genetic mouse data.
Very consistent. Now, they did notice something a bit unexpected in the mouse work.
Ooh, what was that?
The tranexamic acid, it seemed to kind of hang around longer in the system, and it inhibited plasminogen activation for longer than they might have predicted, specifically in the mice that had the surgery to induce the clotting.
Any idea why?
They suspected it might be because the surgery itself could have affected the mice's kidney or liver function a bit, how the body processes the drug.
Ah, okay. But did that affect the main result?
Well, that's the important part, no. Even with the drug present for longer and actively suppressing the system, it still didn't result in larger clots.
Okay, that's key.
And the researchers also noted something else about this specific mouse model. It seems that, well, not much clot breakdown was actually happening within the first 24 hours anyway.
Huh. So maybe inhibiting breakdown didn't matter much for initial clot size, simply because breakdown wasn't a big factor yet in that early phase.
That could be part of the explanation for why inhibiting it didn't change the initial clot size in this specific model, yeah.
Interesting nuance. Okay, let's shift to the big human data sets. The genetics. What did that analysis find?
So in those huge human genetic studies, looking at common variations in the genes for the plasminogen activation pathway proteins, they found these variations were not significantly associated with the risk of venous thromboembolism in the general population.
Not linked at all.
Not significantly. And remember, they compared this to genes like factor V or prothrombin, which are known risk factors, and those showed clear, strong associations, just as expected.
But the plasminogen system genes didn't show that same pattern.
They just didn't. No common genetic fingerprint linking this breakdown system directly to increased vein clot risk in the general population.
Okay, wow. And what about the protein levels, the actual amounts circulating in people's blood?
Also using data from a large human database, the UK Biobank data, They measured the levels of various proteins in this pathway.
And plasminogen itself.
The level of plasminogen protein was not significantly associated with the risk of venous thromboembolism.
Again, consistent. But wait, did any proteins in the pathway show a link?
Ah, yes. This is where it gets a bit more complex, but it's important.
Yeah.
They did find that levels of some other proteins in the pathway were associated with risk.
Which ones?
Specifically, plasminogen activator inhibitor 1, often called PAI, and another one called urokinase plasminogen activator receptor, or UPAR. Levels of those were linked to venous thromboembolism risk.
Okay, that sounds contradictory. If plasminogen itself isn't linked, why would proteins that regulate it or are related to it show a link?
That's a great question, and it points to the nuance. The sources emphasize that these other proteins, PA1 and UPAR, they have really important jobs in the body that go beyond just interacting with plasminogen.
Like what?
Well, they're involved in things like inflammation, how cells signal to each other, tissue remodeling, lots of complex biological processes. So their association with clot risk might actually be tied to those other functions rather than purely their role in the clot breakdown pathway within blood vessels.
Ah, I see. So the system isn't just a simple A activates B, which dissolves C. These other players might be influencing clot risk through totally different mechanisms.
Exactly. They're part of the broader network, absolutely. But their link to venous thromboembolism might be independent of the plasminogen activation pathway's specific job of breaking down fibrin clots inside veins. They might be markers of inflammation or something else that increases risk.
Okay, that makes more sense. So we've got the humans with the natural deficiency, the mouse experiments, both genetic and drug-based, the big human genetic studies, and the protein level analysis. Let's try and tie this all together. What's the big, powerful conclusion coming out of all this?
When you synthesize the findings, looking across all these really different ways of investigating the question, you get a remarkably strong and consistent conclusion.
Which is?
Loss or suppression of plasminogen activation does not appear to increase the risk of venous thromboembolism.
Wow. So that just flies in the face of that long-standing medical worry we started with, doesn't it? The fear that stopping clot breakdown makes dangerous clots more likely.
It absolutely challenges it directly and powerfully. And this has potentially really significant positive implications for patient care.
Oh, so?
Well, one of the lead researchers, Dr. Elisa Wohlberg, she's quoted in the Health News article. She says this research provides reassurance that these bleeding treatments may be safer than previously believed.
Reassurance. That's a huge word in medicine, isn't it? Especially when you're constantly weighing risks and benefits.
It really is. And what it could mean for actual patients, according to Dr. Wohlberg, is that potentially more patients who need these medications might be able to receive them.
Which patients are we talking about? Who might benefit most?
Think about people with severe bleeding disorders, like hemophilia, or trauma patients with major injuries, or even women who suffer from really heavy menstrual bleeding.
Conditions where stopping bleeding is critical.
Exactly. And medications like tranexamic acid, which, as we discussed, suppresses plasminogen activation. While sometimes doctors might have been hesitant to use it or used it really cautiously in these patients because of that fear about causing clots.
Right, that balancing act again.
But this research suggests that fear might be unfounded, at least regarding starting these venous clots. So these treatments might be used more freely, more safely, to prevent severe, even life-threatening bleeding.
That's
Yeah.
potentially huge. Opening up safer, effective treatments for people who really need them. All because of this fundamental research.
It really highlights what Dr. Wohlberg calls the power of discovery science. She makes the point that this kind of basic biomedical research, just trying to understand how the body really works, it can translate directly into better, safer treatments and ultimately save lives.
It's a fantastic example of why challenging assumptions and digging deep into basic biology is so important. Okay, so this is incredibly promising news, provides a lot of reassurance. But, this is the deep dive. We have to look at the nuances, the limitations. No single study answers everything, right?
Absolutely. And the researchers themselves are careful to point these out. First, generalizability. Meaning, while the findings were consistent across these different models and data sets, we can't necessarily say they apply absolutely universally to every single clinical scenario or every patient group, especially maybe patients with really complex health problems or multiple existing risk factors for clotting.
Okay, so it's a strong general finding, but caution is still needed in very specific complex cases. Makes sense.
Exactly. Another important point is what the study focused on. This research primarily looked at whether suppressing the system makes clots form in the first place, specifically venous clots.
Right, the initiation of the clot.
It didn't really dig into whether problems with plasminogen activation might affect how well or how quickly a clot resolves or breaks down after it's already formed.
Ah, that's a key distinction.
Hmm.
So it might not cause the clot to start, but could it maybe affect how long it sticks around or how well it clears up?
Precisely. That's a different question. And there is other research suggesting that related proteins, like having too much of that PAI-1 inhibitor, might actually slow down clot resolution, while maybe having too little speeds it up. So while the system might not be critical for stopping clot starting in veins, it could still play a role in the cleanup process later on.
Okay. And the study also didn't look at longer-term complications after a clot, did it? Like post-thrombotic syndrome.
No, that wasn't the focus here, just the initial formation risk.
Got it. Formation versus resolution. It's
Yeah.
different biological questions.
Exactly. And going back to the mouse models for a second, remember that odd observation about the drug, tranexamic acid, possibly sticking around longer after the surgery?
Yeah.
While main finding held up, that kind of thing, how the experimental setup itself might influence drug behavior, is always something you have to keep in mind when you're thinking about translating animal findings to humans.
Right. Context matters in experiments.
It does. And the human genetic analysis, it focused on common genetic variations, the ones found frequently in the population.
Yep.
It didn't specifically hunt for very, very rare genetic changes, although the researchers do suggest it's probably unlikely that super rare variants would show a strong link to risk if the common ones don't.
Right. Focusing on what's most relevant for most people.
Yeah. And finally, circling back to what we touched on earlier, the complexity, the clotting system is this intricate network.
Definitely not simple.
And the fact that those other proteins, PAI-1 and UPR, did show associations with clot risk, even when plasminogen itself didn't, it just hammers home that these proteins aren't just simple dominoes in one pathway.
They're doing other things.
They have other crucial jobs in inflammation, cell behavior, tissue repair that could influence clot risk completely separately from how they interact with plasminogen for clot breakdown.
It really underscores that you can't just isolate one piece of a complex system like this and predict everything it does, can you? You need to understand the whole network, the other roles these components play.
Which kind of leads us to the bigger picture question that this research raises.
Which is?
Well, if plasminogen activation isn't primarily responsible for preventing clots from forming in veins, what is its main, most critical job?
That's a surprising thought, isn't it? Given how long we've linked it to breaking down clots inside blood vessels.
It is. But based on their findings and other related work, the researchers float the idea that maybe its main role is more about managing fibrin deposition, that clot protein and tissue remodeling outside of blood vessels.
Like in wound healing.
Right.
Or inflammation.
Exactly. Situations where fibrin is involved, but not necessarily inside a major vein causing a dangerous blockage. Maybe its key function is more about tissue cleanup and organization rather than preventing intravascular clots from starting.
So a different primary role than traditionally thought.
Potentially. Or maybe other essential biological processes that we haven't fully appreciated yet, beyond just stopping venous thromboembolism formation. And again, seeing that PAI-1 and UPAIR do link to venous thromboembolism risk, even when plasminogen levels don't, it strengthens this idea that they're independent players influencing risk through those other functions, inflammation, cell signaling, whatever it might be, rather than just by tweaking plasminogen activation.
Fascinating. It really does make you rethink the primary job description for this whole system.
It truly does. So at the end of the day, this research provides really strong, multifaceted evidence that loss or suppression of plasminogen activation does not seem to increase the risk of dangerous clots forming in veins.
Which brings us back to you, the listener. What this means for you is, well, potentially safer treatment options if you or someone you know deals with bleeding conditions.
Yeah, less worry about that specific trade-off.
And it gives us all a clear, evidence-based picture of how this complex system in our bodies actually works, pushing back against a long-held medical concern.
And it opens up new questions, new avenues for research into the true, maybe broader roles these complex proteins play beyond just clot busting inside vessels.
So what stands out to you from this deep dive? Does this finding maybe change how you think about balancing risks in medicine? Or perhaps it just sparks curiosity about what other things we think we know about the body that might be, well, ripe for a deep dive and maybe challenged by new evidence down the line?
Understanding these fundamental processes really is the key to developing better, safer treatments. This study is just a perfect example of that in action.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.